Ready na ba kayo mag-lecture or okay naman? So, this is a very short topic in Junquera. Yan. May yes na ako narinig eh. Kasi if you will read it, it's very short, it's very simple. Kasi skin lang tayo. But anyway, I'd like to ask for your pardon kasi I'm not in my best condition to lecture. Kaya I'm keeping my mask on. And don't worry, hindi naman ako, negative naman ako for COVID. However, you know the respiratory illnesses now, very rampant. So, ako din, I advise you to wear mask while we are in school, no? For your own protection. So, siguro if you will find me parang hinihingal, ayun, sorry for that. Kaya lang, it's better to proceed with the lecture kasi short lang naman siya rather than postpone it. So, I'll go straight, no? For today's topic in histology, it's about the integumentary system or the skin. So, for our learning objectives or what we hope to achieve for this afternoon. Syempre, first and foremost, we have to identify the different layers and sub-layers of the skin. That's the histology proper concept of that. The next is to describe what are the characteristic features, what are the landmark structures and composition of those layers and sub-layers. We will also include in the discussion the different sensory receptors and skin appendages that are also present, no, according to structure, location, and function. And of course, we have correlation, no, of these microscopic structures to their gross anatomic and their corresponding function. So, syempre, our soaking reference will not change with that. It's still Junquera. We included the Fiore kasi we borrowed some images. So, to start with, you know the skin is considered to be the largest gland, single organ of the body. So, this accounts for about 15 to 20% of your total body weight. You can compute for that actually. So in adults, it would be about 1.5 to 2 square meters of surface to the external environment. So since we have correlation with function, let's enumerate what are the functions of our skin and what makes it special. No? What do you think? Una tayo sa pinaka-obvious. Yun, protective function. So it forms a physical barrier against thermal mechanical insults such as friction and against most potential pathogens. So this skin is not just for beautification alone. No, it's our first line of barrier or defense against the environment. And if there could be microbial invasion, there are lymphocytes and antigen-presenting cells that are present in the skin to elicit an immune response. And usually, our resident antigen-presenting cells in the skin will be called what? Diba, we usually have different terms for antigen-presenting cells for different location in the body. So if we're talking of APCs in the skin, we call them yun, Langerhans cells. And of course, the dark pigment melanin, no, in the epidermis protects our cell nuclei from the ultraviolet radiation. It also serves as a permeability barrier against excessive loss or uptake of water. And the selective permeability allows some lipophilic drugs to be administered in our skin via patches, no, skin patches. Another function, no, after protective, na-mention natin, one of the learning objectives. The receptors, no? So sensory. It has a sensory function, no? Many types of sensory receptors allow the skin to constantly monitor the environment. And so, we have various skin mechanoreceptors to help regulate the body's interactions with physical objects because we can sense light touch, we can sense pressure, we can sense different temperatures, cold and hot objects. Okay? The next one will be thermoregulatory. So, a maintenance of a constant body temperature through the skin's insulating components and its mechanism for accelerating heat loss. So, we do have compensatory mechanism wherein we induce fever whenever we have pathologic occurrences inside our body. Ano pa? What other function do we have for the skin? Yun lang ba? Yun, metabolic. What about the metabolic? So there are cells in the skin which actually can synthesize your vitamin D3 that is needed in your calcium metabolism and proper bone formation. So you'll know more about that in biochemistry. Excess electrolytes can be removed in the sweat, and the subcutaneous layer stores a significant form of energy in the appearance of fat. And last but not the least, we have sexual signaling. So many features of the skin, such as pigment and hair, are visual indicators of health and are involved in attraction between genders in all vertebrate species, and that includes humans. Okay, so we have the effects of sex pheromones produced by the apocrine sweat glands and other skin glands that are important for this attraction. So now we are ready to name what are the layers of the skin. Konti lang yan. So can you tell me? Okay. So we have your epidermis and we have your dermis. For dermis, syempre epidermis, they have sublayers. Dermis, it also has sublayers. For dermis, you have your papillary layer and reticular layer. Now we have this subcutaneous layer or hypodermis. Class, this is not included in the major layers of the skin because this is your subcutaneous fascia. No? When you dissect, you actually see your subcutaneous layer or fascia. So you have digitations of the epidermis, such as your epidermal ridges, and projections of your dermis, as you call them dermal papillae. So those two, when they interact together, that's your dermo-epidermal junction. Okay? So let's go to your epidermis, the superficial layer of the skin. So, syempre, when you study for the different types of epithelium, one of the examples, diba, for your stratified squamous keratinizing will be your epidermis, composed of the cells called keratinocytes. Okay. So your epidermis, aside from having keratinocytes, will also have other types of cells, much less abundant. So you have the pigment-producing cells, your melanocytes. You also have what we called earlier kanina, Langerhans cells, your antigen-presenting cells, and your Merkel cells, which are your tactile epithelial cells. Another thing to remember for epidermis before we go to the layers is that we do have a concept of thick and thin skin. So the thickness of the epidermis varies in different locations. Okay? So we have found on the palms of the hands and soles of the feet, you have a thicker skin compared elsewhere on the body. So where does the difference lie, class? What is the difference? What makes a skin thick? Iba yung sinabihan ka ng thick-skinned, ha? Oo, iba yon. Iba din yung histology. So kapag sabihan ka ng thick skin, batuhin mo ng histology. Okay? So, what will be the difference? Yun, naririnig ko na. It lies on the layers of the epidermis. No? So, the thickness of the epidermal layer. So, for thin skin, it could be from 75 to 150 micrometer. And for thick skin, it can go up to 400 to 1,400 micrometer. So there's a certain layer that is present in the thick skin that is not found on your thin skin. So the next illustration from Junquera will be your layers of epidermis in thick skin. So kompleto yung layers pag thick skin. So what are they? From top to bottom. Stratum corneum, stratum lucidum, stratum granulosum, stratum spinosum, and last but not the least, your stratum basale. So among these layers, which will not be found on thin skin? Lucidum. Okay, so take out lucidum, you have your thin skin. So let's describe the composition, the second learning objective, no? Let's start with stratum corneum. So when you view your slide, we do have a very good slide in the laboratory, and I want you to examine each layer intimately, okay? So for stratum corneum, this is about 15 to 20 layers of your squamous keratinized cells filled with your birefringent filamentous keratin. So I already put the bracket for the label of your stratum corneum. And as they form, you have your keratin tonofibrils becoming heavily amassed with filaggrin and other proteins in your keratohyalin granules. So, do you think the cells, because when we say keratinized, it's keratinized already. Do you think the cells here are alive? Hindi na, no? So they are cornified already. So they represent the end of keratinization. So the cells would contain only amorphous fibrillar proteins with plasma membrane surrounded by the lipid-rich layer. And the fully keratinized or cornified cells in stratum corneum will now be called squams. Okay, squams. Which are continuously shed at the epidermal surface as your desmosomes and lipid-rich cell envelopes break down. So, the next layer, we are discussing as to thick skin para complete yung layers, is the single translucent layer called stratum lucidum. Actually, it's not that easy to distinguish in the microscope. But you know that this is only found in thick skin. And a thin translucent layer of flattened eosinophilic keratocytes held together by desmosomes. The nuclear organelles are lost already, and the cytoplasm consists almost exclusively of your packed keratin filaments embedded in your electron-dense matrix. The next layer is, I think, one of the easiest to identify kasi it's the most intensely stained or pigmented in your slide. The stratum granulosum. Okay? So this is about 3 to 5 layers of flattened cells which are still undergoing terminal differentiation process of your keratinization. So I'd like you to remember what are the different structures that could be found here. So the cytoplasm here with your flattened cells with intensely basophilic masses are called your keratohyalin granules. And aside from that, you also have characteristic features which will include Golgi-derived lamellar granules. So these are small ovoid structures with many lamellae containing various lipids and glycolipids. So I'd like to ask the class, what is the significance of this lamellar granules? naka-open ba yung mga ano niya, libro? Yun. Yun. If you have heard the phrase, makes the skin impervious to water. Diba? So, this is it. The last activities of your keratinocytes plus the laminar granules undergoing exocytosis, they produce that lipid-rich, impermeable layer around the cells. And this forms a major part of the skin's barrier against water loss. So please remember that statement. So again, no, together, keratinization and production of the lipid-rich layer have this crucial sealing effect in the skin, forming the barrier to penetration by most foreign materials. I think this is a very significant statement. Kaya naka-underline siya sa akin, okay? The next is, after granulosum is spinosum. So you have to remember this in order. Hindi siya pwedeng kung sinong mauna, no? So stratum spinosum is also known to be the thickest layer among the layers of the epidermis, especially in the area of the epidermal ridges. And it consists of polyhedral cells, as you can see in the illustration, very, very obvious, with central nuclei, with nucleoli pa, and cytoplasm actively synthesizing keratins. So just above the basal layer, now before we go to the basal layer, some cells may still divide, and the junction between your spinosum and your basale will now be called stratum germinativum. Okay? Because in the basal layer, later on, I will show you, you have this intense mitotic activity. So as the cells go up, no? And when they start the process of keratinization, as they go up, you still have some mitotic activity left as you transition to spinosum, and that's the area of your stratum germinativum. So here, the keratin filaments assemble into microscopically visible bundles called tonofibrils that converge and terminate at the numerous desmosomes holding the cell layers together. So this is very important because the cells extend slightly around the tonofibrils on both sides of each desmosome. And this is the reason why it appears to look like short spines or prickles at the cell surface. So aside from being the thickest layer, this is also known as the prickle cell layer of the skin, your spinosum. So the epidermis of thick skin, like your soles of the foot, which are always subject to pressure, no, will have a thicker stratum spinosum with more abundant tonofibrils and desmosomes. So let's add that difference, no? Aside from the appearance of your stratum lucidum, you have a thicker stratum spinosum for your thick skin. Sige, next. Last but not the least, your basal layer. A single layer of basophilic, cuboidal, or columnar cells on the basement membrane at the dermal epidermal junction. So I'd like you to focus on the magnified picture on the right, no, on your right side, bottom side. We have hemidesmosomes in the basal cell membranes joining these cells to the basal lamina. And you have, again, this is characterized by intense mitotic activity, which will have your progenitor cells for all the epidermal layers. As with basal layers in histology, no, they contain the progenitor cells for all epidermal layers. So human epidermis is renewed about every 15 to 30 days, so that depends on the age, region of the body, and other factors. An important feature of all keratinocytes in the stratum basale is your cytoskeletal keratins. And these are your intermediate filaments, about 10 nanometers in diameter. So during differentiation, the cells move upward, and the amount and types of keratin filaments increase until they represent half the total protein in the superficial keratinocytes, until they become fully keratinized or cornified in your stratum corneum. So this is in summary, you have your layers of the epidermis, no? You have your stratum corneum, lucidum for your thick skin, stratum granulosum, stratum spinosum, and then stratum basale. So you will notice parang there's a brownish pigment. Did you notice in the stratum basale? Okay, so that's your melanin pigment. So a closer view of stratum granulosum and stratum lucidum. So C is corneum, L is lucidum, G is granulosum, and S is spinosum. So if you will look at your slides in the scanning, so you have your epidermis, your dermis, and then your hypodermis. And then you have your, if you shift to low power objective, you will still notice the papillary and reticular dermis. So if you want to study for your epidermis, you go to your high power objective, and you can actually see for your stratum corneum, followed by your lucidum, and then granulosum. Just look at the intensely purplish layer, no, with keratohyalin granules, and then your stratum spinosum. And then for stratum basale, just be guided with the melanin pigment and the epidermal ridges. Okay, next. Let's now go to the different cells that can be found in your skin. So we have been talking about the keratinocytes, no, in the composition of the layers. How about the melanocytes? So your melanocytes, the most important factor for the color of our skin. And this is usually found in the basal layer or stratum basale, as you saw in the illustration and pictures in the actual slide. And of course, in the hair follicle. So they produce your black or brown pigment called eumelanin. These are neural crest derivatives that migrate into the embryonic epidermis stratum basale. So one melanocyte accumulates for every 5 or 6 basal keratinocytes, and they would have pale staining rounded cell bodies attached by hemidesmosomes to the basal lamina. They have numerous small mitochondria, short cistern of your rough endoplasmic reticulum, and well-developed Golgi apparatus. So the melanin synthesis of your melanocytes will be initiated by your tyrosinase, which converts tyrosine and then further transformed and polymerized into the different forms of melanin. So your melanin pigment is linked to a matrix structural protein and accumulates in the vesicles until they form mature elliptical granules about 1 micrometer long called melanosomes. So this is where your melanin is accumulated. So I think it's better if we look at the illustration for the next slide. So one melanocyte plus the keratinocytes, which it transfers melanosomes, make up an epidermal melanin unit. So this is your melanosome formation. After it is initiated by your tyrosine, no, in the Golgi apparatus, stage 1, you have vesicle with tyrosinase and other enzymes to form melanin. And then going to your stage 2, melanin production begins on organized matrix in vesicle. Stage 3, melanin pigment accumulates on the vesicular matrix. And stage 4, melanin fills the mature pigment granule. Next are the antigen-presenting cells, your Langerhans cells derived from monocytes. So again, can I ask the class, where do we usually find your melanocytes? Stratum basale. So how about your Langerhans? They represent 2 to 8% of cells in the epidermis. Where do you think are they mostly profound? Yes, so usually clearly seen in your stratum spinosum. They bind, process, and present antigens to your T lymphocytes just as the same manner as any immune dendritic cells in other organs. So the microorganisms cannot penetrate the epidermis without these cells being alerted and triggering an immune response. Now for your Merkel cells, we said a while ago they are your epithelial tactile cells. So these are sensitive mechanoreceptors essential for your light touch sensation. So they are joined by desmosomes to keratinocytes of the basal epidermal layer. So again, where do you find these Merkel cells? Usually in your basal layer. Abundant in highly sensitive skin like your fingertips and bases of some hair follicles. So they originate from the same stem cells as your keratinocytes, characterized by small, Golgi-derived dense core neurosecretory granules containing peptides. Okay? So those are your lesser abundant cells found in your epidermis. Let's go to the next layer, dermis. So this one is made up of your dense, your connective tissue that supports the epidermis and binds it to your subcutaneous tissue or hypodermis. So the thickness of the dermis varies up as to region of the body, and it can be as high as 4 millimeters on the back. So again, they have many projections called your dermal papillae, which interdigitate with your epidermal ridges or epidermal pegs. And that will form your dermal epidermal junction. So what will be the superficial sublayer of your dermis? What do we call it? Papillary. So that's the more superficial of the two. Papillary layer. So this includes your dermal papillae, the projections. And difference between your papillary and your reticular, aside from papillary is the more superficial layer, it's more loose, no? It consists of loose connective tissue with types 1 and 3 collagen fibers, fibroblasts, scattered mast cells, dendritic cells, and leukocytes. So if the connective tissue type is loose, which is more, the cells or the fibers? Hindi pa kayo sigurado. Tapos tayo sa connective. So from this layer, anchoring fibrils of type 7 collagen insert into the basal lamina, helping to bind the dermis to the epidermis. So your reticular layer, this is the thicker, no? So the more superficial is papillary, but the reticular, the bottom layer, is thicker of the two. And it consists of a denser one, no? Dense, irregular connective tissue, mainly bundles of type 1 collagen. So when it's dense, it has more fibers and fewer cells than the papillary layer. So baliktad, it's reversed when it's loose. Okay? A network of elastic fibers is also present, providing elasticity to the skin. So between the collagen and elastic fibers, you also have abundant proteoglycans rich in dermatan sulfate. So this is another illustration which gives you epidermis and the papillary layer and reticular layer of the dermis. So there's really no clear-cut delineation of your papillary and reticular. So when you study your slides, just look for where the connective tissue transforms into a denser one. And syempre, bottom layer yung reticular and thicker. For the dermis, you also have two major plexuses, no, for your blood supply. Microvascular subpapillary plexus between the papillary and reticular dermal layers. And you also have a deeper plexus with larger blood and lymphatic vessels that lies near the dermis and subcutaneous layer. So this is also richly innervated. You have sensory afferent nerve fibers form a network in your papillary dermis and around hair follicles ending at epithelial and dermal receptors. Autonomic effector nerves to dermal sweat glands and smooth muscle fibers in the skin of some areas are your postganglionic fibers of sympathetic ganglia, and there's no parasympathetic innervation present. So this is not included in your layers of the skin, but we have to discuss hypodermis or your subcutaneous layer. This is loose connective tissue that binds your skin to the subjacent organs. You know this very well because you do your actual dissection. It's also called hypodermis or superficial fascia, and they contain your adipocytes that vary in number according to the different body regions and vary in size according to your nutritional state. Extensive vascular supply at the subcutaneous layer, you will promote rapid uptake of your injectionable drugs like insulin. Okay, so sensory receptors. We have diverse sensory receptors that are present in the skin. So they are classified as to they are capsule or encapsulated or unencapsulated. So when they have tissue capsules, they are deemed more complex. For the unencapsulated sensory receptors, we'll be talking about the Merkel cells again. And they function as your tonic receptors for light, touch, and sensing an object's texture. So mind you, when you study for these sensory receptors, you have to take note of their function and location. Free nerve endings, which are found in the papillary dermis, extending into your lower epidermal layers, and they respond primarily to high and low temperatures, pain, and itching, but also function as your tactile receptors. Root hair plexuses, these are web of sensory fibers surrounding the bases of hair follicles in the reticular dermis that detects movement of the hair. Okay, for the encapsulated ones, you have your Meissner's corpuscles. These are your elliptical structures around 30 to 75 micrometer by 50 to 150 micrometer, consisting of sensory axons winding among flattened Schwann cells arranged perpendicular to the epidermis in the dermal papillae. And they initiate impulses when light touch or low frequency stimuli against skin temporarily deform their shape. So numerous in your fingertips, palms, and soles, but decline slowly in number during aging after puberty. Next encapsulated will be your lamellated or Pacinian corpuscle. These are your large oval structures, approximately 0.5 millimeter by 1 millimeter, found deep in the reticular dermis and hypodermis. So they have an outer capsule and 15 to 50 thin concentric lamellae of flattened Schwann cells and collagen, surrounding a highly branched unmyelinated axon. Specialized for sensing coarse touch, pressure, sustained touch, and vibrations. Found in the connective tissue of organs located deep in the body, including the wall of the rectum and urinary bladder. Other examples will be your cross-end bulbs, found primarily in the skin of the penis and clitoris where they sense low-frequency vibrations, and your Ruffini corpuscles, which will have sensory axons stimulated by stretch or twisting in the skin. So the next slides will be about your skin appendages already. So for the hair, this is an elongated keratinized structures that form within epidermal invaginations, which are your hair follicles. So all skin will have at least minimal hair, except for the glabrous skin of the palms. Those are your hairless parts of the body, soles, lips, glans penis, clitoris, and labia minora. So the face will have about 600 hairs per square centimeter, and remainder of the body will also have 60 per square centimeter. For the next slide, I will leave the more detailed reading sa inyo kasi baka mabore naman kayo ng tuluyan. So these are just your parts for the hair. So your growing hair follicle, when you draw it, diba, terminally it will have a dilation. That's your hair bulb. And then a dermal papilla will insert into that base of the hair bulb. Why? Because they contain the capillary network that is required for sustaining or nourishing the hair follicle. Now, the keratinocytes, continues with those of the basal epidermis, cover the dermal papilla, and these cells will form the matrix of your elongating hair root. And the part of the hair that you can see grossly, above the skin surface, is your hair shaft. Okay? See. So you have layers of the hair. We're talking of your hair follicle. In most thick hairs, there are large, vacuolated, and moderately keratinized cells forming the central medulla of the hair root. So that's the innermost. This is surrounded by your cortex, no? Heavily keratinized, densely packed cells make up the cortex around the medulla. And then peripherally pa doon, you have the cuticle, a thin layer of heavily keratinized squamous cells covering the cortex. And the outermost cells of the hair bulb are also continuous with the epithelial root sheath, and which are divided into your internal and external. Internal root sheath surrounds the initial part of the hair root, but degenerates above the level of the attached sebaceous glands, while your external root sheath covers the internal sheath and extends all the way to the epidermis, where it is continuous with the basal and spinous layers. And this is important also. Separating the hair follicle from the dermis is your acellular hyaline layer, and the thickened basement membrane is called the glassy membrane. I think this is better, no? Makikita niyo yung layers. So you have your M, medulla, CO is your cortex, followed by your cuticle peripherally, and then your root sheath, internal root sheath, external root sheath, okay, connective tissue sheath. G is your glassy membrane. So, another structure that is closely related to your hair, it's a smooth muscle that can pull this hair erectly. Ano yung tawag doon? Yun, alam niyo na. So, erector pili muscle. Ay nako, ang hirap mag-discuss when you're not feeling well. Anyway, a small bundle of smooth muscle cells extend from the midpoint of the fibrous sheath to the dermal papillary layer. So, ano lang to, simple as the contraction of this muscle will pull the hair shaft to a more erect position, usually when it is cold, in an effort to trap a layer of warm air near the skin. So in regions where hair is fine, the contraction of your erector pili muscles is seen to produce tiny bumps. Anong tawag natin dito? Layman's term? Yun. When do you feel goosebumps? Okay, so ito yun, yung close relationship nila. So when you examine for your scalp skin, you have there your hair follicle. Adjacent to that, you will find sebaceous glands. And then adjacent to that also, you will see the erector pili. Okay? So yan, ito actual na natin. Scanning of the scalp. No? You have your hair follicle, sebaceous, your sweat, and your erector pili muscle. Anyway, it will also be discussed to you again when you have your lab review with Dr. Glenn. So 3 major phases of hair growth cycle. Let's just define them. No? Anagen, generally long period of mitotic activity and growth. Catagen, brief period of arrested growth and regression of the hair bulb. And then telogen, final long period of inactivity during which the hair may be shed. Kasi diba, we lose our hair, pero bumabalik din naman. No, we have hair growth. So at the beginning of the next anagen phase, epidermal stem cells located in a small bulge of the external root sheath near the erector pili muscle produce progenitor cells for the matrix of a new hair ball. Okay, next naman will be nails. A similar process of keratinization produces the nails, which are the hard plates of keratin on the dorsal surface of each distal phalanx or your fingers. So again, I leave this detailed readings of the different parts of your nail, no, as well as the hair. So the proximal part of the nail is the nail root, covered by a fold of the skin, from which the epidermal stratum corneum extends as your cuticle or eponychium. Ito yung when you do pedicure or manicure, pinapatanggal niyo ba yun? Cuticle, okay. The nail root matures and hardens as the nail plate. And the nail plate that is bound to your epidermis is the nail bed, which contains only basal and spinous epidermal layers. So there's a continuous growth in the matrix that pushes the nail plate forward over the nail bed. If you're curious, the rate of growth is about 3 millimeters per month for fingernails and 1 millimeter per month for toenails. The distal end of the plate becomes free of the nail bed at the epidermal fold called the hyponychium. So this is the slide for your nail. I'd like you to read more on that. Ano to? Chunkera. No? You have your NP is nail plate, no? NR is nail root, NB is nail bed. Okay. So let's go to sebaceous glands. Sebaceous glands embedded in the dermis over most of the body, except a thick glabrous skin of palms and soles. Average of about 100 such glands per square centimeter of skin, and it will increase in the face and scalp. So we have the most number of sebaceous glands in our face and scalp, as much as 400 to 900 per square centimeter. So your sebaceous glands, you were introduced to them prelims pa, diba? So branched acinar glands with several acini converging at a short duct that usually empties in the upper portion of your hair follicle. So the hair follicle plus the adjacent or associated sebaceous gland are called collectively as your pilosebaceous unit. The stem cell niche of the follicle's bulge region also forms the progenitor cells of your associated sebaceous glands. So there are certain hairless regions such as the penis, clitoris, eyelids, and nipples. Sebaceous ducts will open directly into the epidermal surface. And the acini of your sebaceous glands are the classic example of your holocrine secretion. They have a basal layer of flattened epithelial cells on the basal lamina, which proliferate and are displayed centrally, undergoing terminal differentiation as large lipid-producing sebocytes filled with small fat droplets. Okay, so the secretion of sebum is a complex mixture of your lipids that include your wax esters, squalene, cholesterol, and triglycerides hydrolyzed by bacterial enzymes after secretion. So the secretion from sebaceous glands increases greatly at puberty, stimulated primarily by your testosterone in men and ovarian and adrenal androgens in women. So sebum helps maintain the stratum corneum and hair shafts and exerts weak antibacterial and antifungal properties. So this is another illustration of your sebaceous glands from Chunkera. You have there the acini of your sebaceous gland consisting of your sebocytes, the cells, and yung ducts, they open into ducts, and then their secretion goes up into the end of your hair follicle. Next is sweat glands. They develop as long epidermal invaginations embedded in the dermis, and you have your eccrine and apocrine. For eccrine, they are widely distributed in the skin, most numerous on the foot soles. Collectively, there are 3 million eccrine sweat glands of the average person, approximately equal to the mass of a kidney, and they produce as much as 10 liters per day, and that is the secretory rate, far exceeding that of any other exocrine gland. Ganun pala tayo mag-sweat. 10 liters a day. So sweating is a physiologic response to increase body temperature during physical exercise or thermal stress, and of course, the most effective means of temperature regulation of humans. So I'd like to tell you that there are secretory part for your eccrine sweat gland, which is generally more pale staining than the ducts, and it consists of your unusual stratified cuboidal epithelium. And what are the three cell types? So you have your pale staining clear cells located on the basal lamina to produce the sweat, having abundant mitochondria and microvilli to provide large surface areas. You have your dark cells, which are filled with your strongly eosinophilic granules that line most of the lumen and do not contact the basal lamina. The granules here undergo merocrine secretion to release the poorly understood mixture of glycoproteins with bactericidal activity. And your myoepithelial cells, these cells contract to move up the watery secretion to the duct. Okay, so the ducts of your eccrine sweat glands have two layers of more acidophilic cells filled with mitochondria and having cell membranes that are rich in sodium, potassium, ATPase. So these duct cells absorb sodium ions secreted in water to prevent excessive loss of this electrolyte. So at the epidermis, each duct merges with a stratum basale, and sweat flow continues in a spiraling channel through the 5 epidermal strata to an excretory sweat pore in the skin surface. So here's illustration for your eccrine sweat glands. We go to apocrine, which are largely confined to the skin of your axillary and perineal regions. So their development depends on your sex hormones and is not complete and functional until after puberty. So I'd like to emphasize the asini of this secretory component of your apocrine gland will have much larger lumen, and the lining is different. Simple cuboidal eosinophilic cells with numerous secretory granules. I pointed that out, the difference between the lining of your apocrine and eccrine. So thus the glands are misnamed. The secretion is merocrine, not apocrine. The ducts of your apocrine glands are similar to eccrine, but they usually open into your hair follicles at the epidermis and may contain the protein-rich product. The slightly viscous secretion is initially odorless, but may acquire a distinctive odor as a result of bacterial activity. So your apocrine sweat glands are innervated by adrenergic nerve endings, whereas eccrine sweat glands receive cholinergic fibers. So these are your illustration for your apocrine sweat glands. You have your secretory portion of an apocrine sweat gland here. So here is another illustration from the Fiore. So you have there your different layers of your skin, epidermis and dermis, and you have your hair follicles, your sweat glands, adductor pili, and sebaceous. So this is what you will see when you look for the scalp slide. This is under low power objective. You have the hair follicle, the sweat gland, sebaceous gland, and adductor pili muscle. So again, this is in summary of the skin and subcutaneous layers. So again, hindi ito lang yung pag-aaralan. Everything. Kasi short chapter lang naman siya. But anyway, we are done theoretically with the histology of the skin. So, ano yung significance? Why do we need to understand the microscopic structure of the skin? Ano yung clinical application niya? Do you think after studying this, you can forget about this after your exam? So when do we apply this concept? O, tapos na yung practical. Pumasa na kayo ng board exam. Pwede na natin kalimutan. So when do you think this lecture will be applied more frequently than you can think of in your practice? When? Surgery lang? Yun. When you encounter wound healing and repair. Okay? So whether it is deliberate as surgery or basta you have interaction in the skin barrier, you can actually manage it efficiently if you have that good foundation of the histology of the skin. You have that, you know when to remove the sutures. You know when to apply sutures or just leave it to heal by itself. Yung mga ganon. Okay? So for skin repair, the process of cutaneous wound healing, whether it's surgical or accidental, will involve overlapping stages. Meron tayong stages. So in the first phase, of course, you have bleeding. Blood from the cut vessels will start to coagulate in the wound, releasing polypeptide growth factors and chemokines from disintegrating platelets. After which, sinong mga cells ang unang matatap? Neutrophils and macrophage will undergo diapedesis so that they can locally migrate to that area, remove the bacteria and debris from the wound. And this will be your major events of inflammation. Typically, the initial 2 to 3 days of the creation of the wound. Okay, you have inflammation first. After the inflammation, you have epithelialization, which begins as your cells in the basal layer remove their desmosomes and hemidesmosomes, migrate laterally beneath the blood clot, and now will form your desiccated eschar or scabbing. If much epidermis has been lost, new cells may migrate from the bulge region of your surviving hair follicles. And you have the growth of epidermal cells and fibroblasts stimulated by several different growth factors coming from your macrophages and other cells and from their binding sites in your extracellular matrix proteoglycans. Okay? Next, ito yung hallmark. This is the hallmark of wound healing, the formation of your granulation tissue. You have the proliferating fibroblasts and newly sprouted capillaries. This is when you establish new blood supply for that area. So you have a well-vascularized tissue already, which is called your granulation tissue, that will replace the blood clot. And in the final stage, the epidermis will reestablish the continuity of the skin, but has lost the ability to form the new hair or glands. Now, the granulation tissue now will undergo remodeling, and a more normal vasculature is reestablished. So, collagen bundles and fibroblasts in this new connective tissue are at least initially much more abundant and disorganized than in uninjured skin, producing scar tissue at the wound site. So, these are the different major stages of your cutaneous wound healing. So first, you have cut blood vessels bleeding into the wound. The second, you have the initiation of your inflammation, where leukocytes will migrate to the blood clot. Then blood vessels regrow, and that's the formation of your granulation tissue. After which, epithelium regenerates, your connective tissue will have fibrosis, and then it will now, the granulation tissue will undergo remodeling. So you will know more about this in your higher years in pathology and surgery. Okay, so ano yung next slide natin?
Thank you.
Merry Christmas!